Hey guys, so greetings from nighttime, Mrs. Morse. Okay, so we're going to talk today, or we're going to finish like what we were talking about yesterday. So we all covered this part of the video for the photosynthesis review and the one about the electromagnetic spectrum. And if you do not have your picture of the electromagnetic spectrum, and the chloroplast, you should go get that now because we're going to use that. Preferably with colored pencils. The good deal about this is that you can pause this video, and you should pause this video to continue on your picture of photosynthesis, and then we're going to continue on and draw the light and the light dependent and the light independent reactions. Okay, so moving on to our second video, which really talks about kind of the first part of photosynthesis. So take a second. I'm going to make this bigger. Pause this and go through and make sure that you have the right answers. And then we're going to use them to work on our picture. So I assume that you just did that. Sorry, I'm having cat problems. So where does this happen? So the light reactions and the Calvin cycle. And so just for some context, the light dependent reactions are sometimes called the light reactions. So we'll use those terms interchangeably. They also involve electron transport chains, which is something that sometimes you'll hear those terms, but I'm going to try to call them the light or the light dependent reactions every time I talk to them, but they have a couple names. They happen in those thylakoids that we talked about before, and it involves a bunch of different proteins that essentially get energy from the sun and use it to make ATP and NADPH. That's like the big thing. The other big thing that happens in this step is that you split water. Water is broken apart into its pieces. Remember that we covered water extensively earlier in the year, I think, all of us, maybe even. Sorry, again, cat problems. So without further ado, let's go and look at this and draw our picture. So bear with me while I switch computers. Okay, so here we are back at our picture, and we are going to draw these light reactions. I'm going to draw mine underneath the electromagnetic spectrum here. So that's where I'm going to make it. And so I'm going to take, essentially just do one of these things. And I'm going to draw one giant thylakoid, and then I will, because we're just gonna, and I'm leaving a gap in mine because that's where my ATP synthase is going to go. Okay, so we are going to call this the light dependent reactions of photosynthesis. This is apparently going to be the picture where I never write photosynthesis properly. Okay, so here in our thylakoid, you have photosystems. You have two of them, and they're made up of a bunch of different pigments that hang out in the thylakoid membrane. Mostly chlorophyll, but some accessory pigments too, like those carotenoids and those phycocyanins. So let's take our green. Nope, this is bad. I try to do the thing where I throw away the markers when they don't work, but I do a lot of sharing of these markers. All right. Okay, so what happens is you have two photosystems and they're called photosystem 2 and photosystem, that looks like an A, but it's not, photosystem 1. And we start at photosystem 2. Remember, that's because it was discovered first, not because of anything logical, like it happens first. Photosystem 2 comes first. Sorry about that. I didn't invent it. But you have photons, which are essentially particles of light from the sun. It doesn't really have to be sun. It can be from a light bulb or some other kind of incandescent light. Plants can use those. You can buy grow lights and things that are even better than that. But essentially what happens is that in photosystem 2, you excite electrons. Those photons make, and remember that electrons are like our little letter E's. And electrons are going to get passed around from these little photosystems. And I don't think the video did a great job, but they get passed around and eventually they get a bunch of energy. They get to one special pigment, and that pigment is going to take that energy collected from all of these pigments, and they're going to use that energy to split water. So, and it takes, so I'm going to take a marker now because I think that it's time. And it is going to take water and split it. So water is going to turn into oxygen gas, which is going to just float away and become used for cellular respiration, or remember your leaf cross section is going to be released through the leaf through the stomata. So that's what happens to the oxygen. Now you're left with hydrogens. Hydrogens are the smallest atoms. They have 1 electron and 1 proton, and that's it. And so the electrons are going to go back to replace these guys. And the protons, which are just H pluses, end up inside the thylakoid. And so really what this is it's like, let's cut a little hole in this. So we're looking inside the thylakoid. And so this keeps happening. There's also a pump that does this. But in the end, you just end up with a whole mess of hydrogen ions inside the thylakoid. Cool? Cool. Okay. These electrons are going to end up down here at photosystem 1. And photosystem 1 is going to use photons. And actually, they're energetic. Let's highlight our photons. So we're going to get new photons, again, from our light. I have to ask my trusty cat friend Bo here to move in a second. So more light excites the electrons are here. So if you didn't have electrons here and you don't have any water to split, you can't continue this process. So if there's no water, then there's no electrons here when you split from splitting water. So you can't make this process happen. So no water, no photosynthesis. No light, no photosynthesis. That's kind of how it works. So here you go. You have your electrons that are here from your photosystem 2. There's light, so they're going to bounce around. And then You're going to get to one special one. We'll put it on the end because that's most convenient. And that part of the photosystem is going to bounce it up in energy, and it's going to use that energy to take NADH plus, it's going to stick those, sorry, NADP, sorry, I'm just in the wrong photosystem, sorry, in the wrong process. Take NADP and make it into NADPH. Hold on, we need to move the cat. Can't really tell because he's almost paper colored, but come on, bud. Don't bite me. There we go. Okay, so you're gonna take your NADP and you're gonna stick some hydrogens on there. We're gonna find out where the hydrogens come from in a second, but the electrons are coming from here. So you're gonna make NADPH. Okay, so here's our product. Let's color code some stuff. Let's take some colors we've not used much. Not used blue and pink. Let's use blue and pink. Why not? Okay. So we're going to take a pink. I'm going to put a pink box around the products of photosynthesis. Oh, let's say just products. And I was going to use red for reactants, but that doesn't really make sense. We'll use blue for reactants. Okay, so products are pink. Blue is reactants. Let's do, my violet isn't very good. I don't think, maybe. Oh no, this violet's good. We're going to do violets for high energy molecules, intermediates, and we'll use sort of the seafoam green for low energy intermediates. And by intermediates, I mean things that we recycle and we use over and over again. I think you'll see in a second. Okay, so let's just look at what we've done. Nope, let's do one more thing. So in this gap, there is an enzyme in the membrane called ATP synthase. And it's a protein, and it is essentially, it's like a gate. It's like a turnstile. If you've ever been on a subway, the turnstile is the thing that you walk through and it flips and it counts that you've been through. And essentially what happens is that every time it lets a hydrogen out, it turns over and it uses that to create energy, to harness the energy of those hydrogens. Here's the deal. These hydrogens want desperately to get out of this thylakoid. They want to do that because there's this thing called diffusion where all the molecules that are similar and stuck together want to spread out from each other. And there's a second thing that happens, that when they're all packed in there and they're all positively charged, they repel each other. So all these hydrogens are like really, really, really unhappy here, and all they want to do is leave. And so the ATP synthase is the way that that can happen. So what the ATP synthase is going to do is let these hydrogens out. And when it does that, it turns that turnstile, if you will, and it takes ADP plus P, because ADP is adenosine diphosphate, and it uses the energy from letting the hydrogen out to make that into ATP, adenosine triphosphate. It takes this phosphate and sticks it on there. So ADP into ATP. Okay, so, and it does that a lot, like as long as there are hydrogens in there, which is to say as long as there's both light and water and the light reactions are happening. So let's use our markers to label our stuff. So reactants for the product. So remember that our photosynthesis reaction, photosynthesis is, you can see that, right? Yep. Photosynthesis reaction is 6 water plus 6 carbon dioxide. As long as you have light, you will end up with 1 glucose plus 6 oxygen. Okay, so, oops. Cool. So let's take that information and use that to label any reactants and products we have here from the full reaction. So reactants, here's your water. So this is where the water comes in. And it creates the product of oxygen. But some of the big deal things is that you take low energy intermediates, ADP, and a phosphate, and NADP, and you make them into high energy intermediates, ATP and NADPH. And the H's, the hydrogens, actually come from these hydrogens that release here. The photosystem 1 takes the electrons and the hydrogens and sticks them on the end here and makes NADPHs. And so ATP and NADPH are both really high energy intermediate molecules. They can't hang around for a long time. They're pretty unstable. But ATP can be used to do cell work. And in the next step, we're going to use it to make carbohydrates. All right, but before that, we're going to go back and look at our key. So that is all of that. Oh, there are a few more. All right, so take a second, pause the video, go through and make sure that you have the answers to all these. But that's loses, not looses, loses. Okay, cool. And this is a summary of what we just did. So hopefully that makes sense to you. All right, now let's get down to the Calvin cycle. So we are going to talk about the Calvin cycle. Next, the Calvin cycle takes place in the stroma, which is the space in the chloroplast that does not have the thylakoids. I'll show you on our chloroplast in a second. And this is the place that takes carbon dioxide out of the atmosphere and makes it into carbohydrates. It doesn't quite make it into glucose. There's another reaction that makes it into glucose eventually, but you get really close to that. So you're making carbohydrates. You're putting energy into bonds between carbons. So let's make that happen. So again, pause the video, make sure that you have the answers to this, and then we'll draw it. So I'll meet you at my other computer. Okay, so zooming back out, looking at, we're gonna be over here, our chloroplast, those were the light dependent reactions. And our next set is called our Calvin cycle, or the light independent reactions. of photosynthesis. Almost did it again. Okay, so sorry about that. Okay, so all of this takes place in the stroma. So inside your chloroplasts, you have thylakoids stacked in grana. One of them's a granum, but whatever. And then the stroma is all the space that's not there. And the stroma is where the Calvin cycle happens. So the Calvin cycle is called a cycle because it's a circle. It doesn't really begin or end, but it kind of begins. Not bad if I do say so myself. This is an incredibly complex process that I'm going to simplify into 3 little steps that are going to be pretty straightforward. Ooh, spoke too soon. Did not do a great job dividing that into 3. Could have done better. All right. So our first step is carbon fixation. In carbon fixation, you start with a molecule called RUBP. I don't really care that you know that, but it's a type of carbohydrate. And you add carbon dioxide. It is where your carbon dioxide enters the carbon cycle. And the important thing is that you know that carbon fixation makes carbon dioxide organic, meaning that it binds it to other carbons and stores energy in that molecule. All right, so carbon dioxide comes in, it binds to RUBP. Now you have carbon dioxide being organic, and that's like a really big deal because that's what you want out of this process. The next step is called reduction. And in reduction, you add energy. To the bonds. Okay, and what kind of energy? The energy comes in the form of ATP is invested. So the energy comes out of it and it becomes ADP plus inorganic phosphate, and NADPH becomes NADP. Okay, so where did the ATP and the NADPH come from? Hopefully you're seeing, I need a pencil, that they're coming from over here. So the ATP is coming from there. The NADPH is coming from photosystem 1. That's where they come from. And so we'll answer where these intermediates go in a second. So now at the end of reduction, you end up with a carbohydrate. If you run the Calvin cycle 6 times with 6 different carbon dioxides, then you end up with a glucose. You need to do some finagling, but you end up with a carbohydrate. So we'll say 6 times equals 1C6H12O6, which is your glucose. All right. After reduction, you have regeneration. And that's when you recreate the RUBP. It's a lot of work, but actually it's a big deal because you can't do that. So it's kind of a big deal. So then you make the RUBP. There's actually lots of them. It's very complicated. But again, you have the RUBP. You can add another carbon dioxide. That's fixation. Then you add energy in the reduction step. You add energy in the form of ATP and NADPH, and then you are going to regenerate it. It actually costs some ATP to make that happen too, but we're not going to talk about that so much. So let's go back. Let's find our reactants. So our reactants, remember that carbon dioxide is a reactant. And glucose is a product. And so now we have our whole reaction covered. You have water and carbon dioxide coming in, and light, which remember isn't a reactant, but is an energy source that is necessary for the process to continue. And then you have oxygen and glucose. So now we have all of those. We also have our intermediates. So you start with your high energy ATP, which came from the ATP synthase, which was generated technically from photosystem 2, pumping the hydrogen ions from the water it split into the thylakoid. And when it's used in the process of the Calvin cycle, it creates low energy ADP and 1P, 1-phosphate, and those are going to go back. Do I have another colored pencil? I don't think I do. Those guys are going to go back to the thylakoid. Unfortunately, the thylakoid is right here. So the Calvin cycle is happening like right here. The light reactions are happening here and the Calvin cycle is happening here. The ATP and the NADPH are going here. And then the ADP and the NADP are coming back, and they're just going back and forth like that. Does that kind of make sense? And you guys did your gizmo on that. All right, so same thing. Where did my button go? Your NADPH is coming in. That's your high energy molecule. And you're using the energy and you end up with NADP. The hydrogens got stuck in the carbohydrates. So that's actually where these hydrogens come from. Cool. Cool. All right, you guys. So not this detailed for your quiz, just summaries. I would know, you get to use your notes. So I would understand what this means. I would watch this video a few times. I think that makes sense. Keep an eye on your paper. Your quiz is tomorrow. It has information on going as far back as the POGO that talks about like what's in a leaf, the leaf cross section, and then you should know a little bit about the light reactions, a little bit about the light independent reaction of the Calvin cycle. And so that's it. You can use your notes and you get to take it twice. So low pressure. And I look forward to seeing you guys tomorrow. So make sure you stay in the Zoom until the end of the period and you're dismissed. But you can always come back and watch this for a refresher later. All right, bye y'all.